Lipid Peroxide Fluorometric Assay Kit. Fluorescence Microplate Reader Experimental Guide. Elabscience Lipid Peroxide Fluorometric Assay Kit. 1. Suitable for LPO assay of suspension cell and adherent cell. 2. This video shows the process using fluorescence microplate reader for suspension cell detection. Unpack the kit, check the label and items. Read the full instruction before use. Please contact the supplier with any question. Reagent preparation. Preparation of buffer working solution. Mix well 5 ml of double distilled water and 5 ml of buffer. Mix fully. 5 micromoles per liter probe solution. Mix well. 4,990. 5 microliter of buffer working solution and 5 microliter of 5 millimoles per liter probe. Mix fully. Protected from light, and it should be used up within 2 hours. 100 micromoles per liter positive control solution. Mix well. 4,995 microliter of buffer working solution and 5 microliter of 100 millimoles per liter positive control. Mix fully, protected from light, and it should be used up within 2 hours. Sample preparation. Plate setting. Operation process. Collecting cell. Transfer cells to the new EP tube. Centrifuge at 300 times of gravity for 5 minutes and collect cells. The collected cells are resuspended with the appropriate working fluid. Add probe working solution to control, positive control and experiment tube. Add buffer working solution to blank tube. Incubate the cells at 37 degrees with shading light for 60 minutes. Centrifuge at 300 times of gravity for 5 minutes. Discard the supernatant. Add 0.5 milliliter of buffer working solution to resuspended the cells. Centrifuge at 300 times of gravity for 5 minutes. Discard the supernatant and repeated 2 to 3 times. Measurement. Add 100 to 200 microliter buffer working solution to resuspend cells. Transfer to the 96 Wells black microplate for measurement. Fluorescence microplate reader instrument parameters: excitation 500 nanometers, emission 540 nanometers.